Okay, so let's talk a little more about isolating a microbial species. To isolate certain species of microbe, you have to consider the needs of the species you are trying to isolate. Now, we talked about this at the very beginning of this lecture series. Let's bring it back around full circle. So you want to consider nutrients, and so that's going to be looking at your building block sources. So what's the stoichiometric ratios of nutrients that the microbes need? How much nitrogen does this microbe need? How much phosphorus does this particular microbe need? How much sulfur does this particular microbe need? Does it need any growth factors like vitamins? Does it have micronutrient requirements that are unusual? So those are the nutrient considerations you'd have for to isolate a particular microbe. Energy source. Does it need nutrients in the media for energy, or does it need light? What does this particular microbe that you're trying to culture use for energy? Optimum oxygen availability, right? Because microbes that are not tolerant of oxygen will need to grow anaerobically. One way is to use a medium that reduces oxygen into something else. Like there are certain media that you can use that changes oxygen into water. And then there's less oxygen in the plate, in the petri dish. But there are also several ways of incubating media and bacteria in anaerobic chambers. So you might need to remove oxygen or create an anaerobic chamber in order to grow certain microbial species. You also would want to know if the particular microbe that you're interested in is abundant or rare. If the bacteria are very rare in the original sample, there are ways to increase the numbers of bacteria so you can work with them more easily. You can run, for example, water with bacteria through a filter so that lots of bacteria accumulate on the filter, and then you put that filter onto the media. You can also use enrichment culture that's designed to encourage the growth of a rare species if you're interested in a rare species. Conversely, if bacteria are too abundant to count easily, so that's another thing. You might not think that having the bacteria be too abundant is bad, but if you think that, you just haven't had to count microbes under a microscope before. So if you have a petri dish or even a slide that is just filled with hundreds and hundreds or even thousands of bacteria, it is really, really hard to count them. And counting them can be important to figuring out how well a particular antibiotic works. So sometimes if a particular microbe is too abundant, it's difficult to get accurate counts, which means it's difficult to measure effect. So you might actually want to reduce the number a little bit and get that sweet spot between rare and abundant. So there are sorry, I lost my space in my notes here. So if bacteria are too abundant to count easily, you can perform serial dilutions of the original sample, sort of like homeopathy, if you know how that works. But this is with bacteria instead of plants, right? It's not an advocation for homeopathy. I'm just saying that it's the same idea in terms of what you do. So you don't do it until there's no microbe left, right? Which is what you do with homeopathy. You do it until there's nothing, no plant in it left. But this idea is you take, like let's say you have 100 milliliters of microbe, of a water mixed with microbe. So you mix that up, then you take a pipette and you take out 1 milliliter and mix it in 90 milliliters of water. Then you mix it, and then you take 1 milliliter of that and put or 10 milliliters of that and put in another 90 milliliters of water, or so on and so forth. So you 10 milliliters to 90, 10 milliliters to 90. That's a serial dilution. You can keep doing that to get the number of microbes lower and lower and lower until they're easier to work with. Let's see here. All right, if there are many other kinds of bacteria in the same sample, you can narrow down the species to the one you're interested in by using what's called selective media. Selective media is designed to be poisonous or unfavorable to all types of bacteria except the one you want. So for you to be able to do that, you need to know something about your bacteria, right? You need to know what makes it different from the other bacteria in the same community. So let's say you took a soil sample, there's hundreds of species in that soil, you're only interested in one. You have to know enough about that bacteria to know what it likes that the other species of bacteria in there don't like. So maybe you'll deprive maybe it has a unique nutrient need, or maybe there's a toxin that it's particularly resistant to, or whatever it is. So you can use selective media to favor the growth of the one you're interested in and stymie the growth of the others. You can also use differential media, which helps the species you're looking for show up in an obvious way. For example, some species of bacteria might phosphoresce if you give them a certain substrate. So selective media will favor the growth of your microbe and disfavor the growth of other microbes. Differential media will make the microbe show up in an obvious way so it's easier to see in a mixed plate, like causing it to glow in the dark or something like that. So I want to talk about Koch's postulates now that we've talked a little bit about microbial ecology. You'll remember these are Koch's postulates. Robert Koch came up with these as he helped figure out germ theory to figure out the microbial cause of a particular disease. So there were all these diseases. People were just figuring out that microbes could be the cause of at least many of them, if not all of them. So how do you find out which microbe is the cause of disease, or if a microbe is the cause of disease? Well, Robert Koch came up with these postulates to figure it out. Oops, this goes on automatically for some reason. I want to start bringing to your attention that many microbes, it turns out, don't fit neatly into these postulates, even though they cause disease. And I'm going to give you one example, and we'll talk about other examples as the course progresses. So I'll remind you that Koch's postulates, the first criteria for identifying a microbe as the cause of disease is that microbe has to be found in all cases of disease and not found in healthy individuals. Second criteria, the microbe can be isolated from a diseased animal or patient and maintained in pure culture. 3, healthy animals will contract the disease when inoculated with the isolated microorganism that you took from the sick animal, even after several rounds of passage between animal and culture. 4, the microbes isolated from the newly sick animal should look exactly the same as the original microorganism that made the original animal sick. Okay. Now, we talked this class about how few microbes can actually be cultivated in pure culture, right? We talked about how there are 12 phylum of bacteria for which at least one species in that phylum has been cultured in a lab. But there are 72 candidate phyla for which no individuals have been cultured in the lab, even though we know that they exist. So these 2 points on Koch's postulates, you really have to take these with a grain of salt, right? Because just because you can't culture something in a petri dish does not mean it's not a pathogen. Doesn't mean it doesn't cause disease. All it means is it won't grow in a petri dish, right? So let's look at an example. This is a picture of a microbe called Pneumocystis jurovecii. Pneumocystis jurovecii. Pneumocystis is a genus of protists. Well, we thought they were protists for a long time. So Pneumocystis species were thought to be protists for a long time. But DNA sequencing now reveals their similarity to single-celled fungal groups. So they might be more like a very primitive fungus. Each species in this genus colonizes a very specific animal species. This particular species shown here, Pneumocystis jurovecii, colonizes the human lung and is responsible for pneumocystis pneumonia, sometimes known as PCP, which is often fatal. One reason this species of microbe is so poorly understood, actually this whole genus of microbe is so poorly understood, is because it cannot be maintained in culture. It is an obligate biotroph, which means it does not live outside of its living host. And colonies do not, so that means colonies do not persist outside the host. They cannot be cultured in a lab. DNA sequencing of the genes it contains is the only method we have for studying it currently, because it's very difficult to observe it while it's still inside a living patient. So I just want to bring that to your attention, that these 2 points of Koch's postulates really have to be taken with some salt. We know this is a pathogenic organism. We know that it causes disease and even death in human individuals, and yet we can't culture it in a lab. It's still a pathogen. So we'll talk about these other points in Koch's postulates also need to be taken with some salt, but we'll talk about that more as the term progresses.